Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about a very important topic of genetics, that is Lampbrush chromosome. Okay, and this video is going to be useful for BSc and MSc zoology students as well as CSIR NEET aspirants and also for NEET aspirants. So without further delay, let's begin. So see, Lampbrush chromosome is a type of giant chromosome. Okay, we know that polytene chromosome is another type of giant chromosome. So as these are the giant chromosomes, they can be easily seen under the light microscope. Okay. Now, this Lampbrush chromosome was first observed by scientist Fleming in 1982 in amphibian oocytes. Okay, so this is very important. This type of chromosome is found in oocytes. Okay, and it was first observed in amphibian oocytes. But you have to remember that not only in amphibians, they are found in many other animals' oocytes too. Okay, but except mammals. Okay, so in mammals Lampbrush chromosome is not found. You have to remember this. It is very important to remember. Okay, now another question that may come to your mind, that during which stage of cell cycle this chromosome can be seen. So this chromosome can be seen during diplotin stage of prophase 1 of the meiosis in the oocyte. Okay, so during oogenesis, this stage is seen during diplotin stage of prophase 1 of meiosis. Okay. Now, this Lampbrush chromosome is actually a meiotic bivalent. Okay. Now, what does the word meiotic bivalent means? Meiotic bivalent means we know that during meiosis, two homologous chromosomes with their sister chromosomes, okay, so sister chromatids pair up. Okay. So see, this is so let me draw it with another color. So this is one chromosome, okay, and let me draw it with another color like green, and it is another chromosome, okay, and these two are homologous chromosome. Okay, these two are homologous chromosome. Now you can see the two homologous chromosomes are crossing each other at a point and they are making an X-like structure, and this X-like structure is known as chiasmata. Okay, so see here, the blue one is the paternal chromosome here, whereas the pink one is the maternal chromosome here, and these are the homologous pair of chromosomes. Okay, so they are meiotic bivalents, and at the places where they cross each other or where they are holding each other together, that place is known as chiasmata. And see here, the place looks like a X. Okay, looks like an X. Okay, now though we are seeing that the paternal chromosome is like a single strand and maternal chromosome is like a single strand, but actually they are not. If we zoom into the maternal chromosome, we can see that there is a structure like this, and when we zoom further, we can see that it is actually made up of two sister chromatids. Okay, so maternal chromosome is made up of two sister chromatids, and similarly the paternal chromosome is also made up of two sister chromatids. Okay, and the interesting thing is that this sister chromatids is not similar in its structure in all the places. Okay, in some places they are forming large lateral loops like this. Okay, so these are the large lateral loops of the sister chromatids at some sites. Okay, and some sites they are forming small loops or more condensed structures. So these loops are known as chromatin loop, and these more condensed structures are known as chromomeres. Okay, so these are the chromomeres and these are the chromatin loops. Okay, these are the chromatin loops. You can see this is from the maternal chromosome part and the blue one is from the paternal chromosome part. So you can see here the Lampbrush chromosome is actually a meiotic bivalent consisting of two pair of sister chromatids. That means four sister chromatids, because two sister chromatid from paternal chromosome and two from maternal chromosome. So there are total four sister chromatids or two pair of sister chromatids held together by the chiasmata. Okay, or chiasma, whatever. So, and at certain places the lateral loops are formed by the unfolding. Okay, so these lateral loops are actually formed by the unfolding of the sister chromatids. Okay, and you have to remember that whenever there is unfolding of any part of chromosome or chromatin, that means they are the active sites of transcription. Okay, we know that when RNA is formed from the DNA, the DNA needs to be unfolded, okay, so that RNA could be formed very easily. Okay, so these lateral loops, these chromatin loop or these lateral loops are unfolded structure of chromatin or chromatid, sister chromatids. Okay, and they are actually the active sites of transcription. Got it? Active sites of transcription. Now you may ask me why there are so many lateral loops in Lampbrush chromosome. Okay, in normal meiotic chromosome we cannot see so many lateral loops, because you have to remember that these Lampbrush chromosome is mainly found in oocyte. Okay, so where the eggs are forming, oogenesis is occurring. Okay, and after that these eggs will combine with the sperm, will be fertilized by the sperm, and zygote will form. So this whole process of early embryogenesis, oogenesis and then early embryogenesis requires different types of proteins, and to form those proteins by the process of translation you need different types of mRNAs, okay, respective mRNAs, and for that you need to transcribe your DNA. So for that reason there are so many lateral loops, so that so many transcriptions can occur at the same time and so many proteins can be delivered during oogenesis. Okay, because it is a very active phase in any organism's life. Okay, so now another question may arise in your mind, why this chromosome is known as Lampbrush chromosome. See, in this structure you can see that due to presence of several lateral loops or chromatid loops or chromatin loops of both the paternal and maternal chromosome, the overall structure looks like a lamp cleaning brush. Okay, we know that a lamp cleaning brush looks like this. Okay, if you have a lamp cleaning brush in your house you can see that. Okay, so the overall structure looks like a lamp cleaning brush. That is why this chromosome is known as Lampbrush chromosome. So see here, due to formation of several large lateral loops from each sister chromatids of two homologous chromosomes, the overall structure of the chromosome seems like a lamp cleaning brush. That is why it is known as Lampbrush chromosome. Okay, so I hope the thing is clear in your mind. What is Lampbrush chromosome, where was it found, okay, and why it is called Lampbrush chromosome, and what is its structure actually. Okay, so if you find this video useful, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.